Assalamu alaikum dear students. Today we will discuss about the formation of phosphodiester bond in polynucleotides. Now a nucleotide that is formed by three basic components. One of the component is called as what nitrogenous base. It can be either a double ring structure or single ring structures. Double ring structures are nitrogenous bases. They are called as purines, while single ringed nitrogenous bases, they are called as what pyrimidines. Now adenine and guanine, they are double ringed purine nitrogenous bases, and cytosine, thymine or uracil, they are purines or single ring nitrogenous bases. Second component is what ribose sugar in RNA, and deoxyribose sugar in RNA. In deoxyribose sugar, this oxygen that is less as compared to what ribose sugar. And you are familiar with the five member structure or furanose form of the ribose already. And then this is what phosphoric acid, molecular formula of phosphoric acid is H3PO4, and it is like phosphate group that is attached at one side with oxygen, and on rest of the three sides hydroxyl groups, they are present. Now how these components, they are combined in a nucleotide. This is a purine nucleotide, or in nitrogenous bases adenine and guanine, they can be there. Always nitrogenous base with its double in its double ring structure with its five-sided structure, it makes a bond with what? With ribose sugar. Like you can see, this is what five-sided ring, and this one is what six-sided ring. Now and with its carbon number 5 phosphate group in the form of phosphoric acid that will make a bond with it. And this hydroxyl group of the phosphate group and hydroxyl group of the carbon number 5, they will make a bond like hydrogen from the phosphoric acid and hydroxyl from the ribose sugar, that will get removed, and as a result a bond that gets developed between the carbon number 5 of the ribose sugar and oxygen of the phosphate group. This bond is called as CO bond or ester bond. This is the first ester bond which is formed within the nucleotide, between the phosphate group and that of the ribose sugar. Now if I draw another nitrogenous base, another nucleotide, then it will be in this form, CH2, and maybe it is a single ring structure. I have to make a bond from this side. Okay, now this can be either cytosine or uracil, because I am drawing but ribose sugar, so definitely it will be but uracil. Here hydrogen, that will be there, hydrogen, hydroxyl group, hydrogen, hydroxyl group, this here will be hydrogen, and from a hydroxyl group from this carbon, that will get removed. This is carbon number 1 2 3 4, and this is carbon number 5, and then a link that will develop between the phosphate group and this ribose sugar. So as I told you earlier, this CO bond, that is called as what? Ester bond. and this is the first ester bond which is formed, and then a hydroxyl group of the next nucleotide that is present at the with the 3 carbon, and hydrogen from this phosphoric acid that will get removed. As a result, another water molecule that will remove to make a second ester bond. Second ester bond means first ester bond that is formed within a nucleotide, and second is formed between two nucleotides. As these two ester bonds, they are connected with the help of phosphate group. That's why we call this bond as what? Phosphodiester bond. And still you can see 5 side that is free to attract any incoming nucleotide, and at this end a 3 side, that 3 carbon, third carbon that is free to attract any incoming nucleotide. That's why chain that will be from 5 prime to 3 prime. Always we name the chain from the 5 prime to 3 prime. So if I write it in a neat form, then it will be like this. So if I draw it in a complete form, then it will be like this. So like, then it will be clear to you that how a diester bond that is there. It can be any kind of base. Then CH2, this is carbon number 5, linked with the phosphoric acid, in this pattern, and hydrogen, hydrogen, hydroxyl, hydrogen, hydroxyl, and here hydrogen that will be there. Now as I have told you that when next nucleotide that combines with this, then this hydroxyl group of the carbon number 3 that gets removed, and it makes a link with the oxygen of the phosphate group, with the oxygen of the phosphate group. So OCH2 and C hydrogen. Let me complete this sugar molecule, which is ribose sugar. And here any nitrogenous base that is there. Now students, you can see that there is a CO bond which is called as ester bond, which is connecting two adjacent nucleotides together, and another CO bond that is present within the nucleotide, which is making a link between ribose sugar and the phosphate group. So as two ester bonds they are there. This is the first ester bond, and this is the second ester bond, which is linked with what? Linked through what? Phosphate group together. That's why this bond is called as what? Phosphodiester bond. And you can see that now two nucleotides, they are linked together, and still like this 5 end and this 3 end. This is carbon number 1, 2, 3, 4 and 5. This 3 and that is free to attract any incoming nucleotide to make a bond with it. That's why we say that either it's DNA or RNA, they are having 5 primes and 3 primes, and they are named from the 5 prime to 3 prime. Like if this is a adenine nucleotide, so it will be named from here, and then if this is a uracil nucleotide, then it will be called as what, uracil. Then the second nucleotide, that will be what, uracil. So this is all about phosphodiester bond during the formation of what, polynucleotides. Thank you, students.